Okay, hello everyone. So next day, last day, the workshop. First of all, let's see if you have any questions from
Crowd room.
Nobody? So you were supposed to get the script which we'll use in today's class. Okay, so do you have any problems with installing Surat? I know that some of you have. So I think you can try to use one of those computers. Okay, so today we'll basically analyze some data sets using Surat. And I'm proposing to start with the SmartSeq2 dataset. Okay, so first of all, let's download the dataset. So this is dataset from a paper which was published recently in Science, Developmental and Oncogenic Programs in H3K27M gliomas dissected by single-cell RNA-seq. Okay, so it's a couple of thousand cells. I'm not sure how many, but when we analyze the data, we will see. So, okay. This was what? Case study where they did some biological inference. It was samples were collected at the time of surgery. So if you're interested in the details, you can either read the paper or let's see what's done here. So they had some full-length single-cell RNA-seq, right? Basically, it was SmartSeq2 protocol, and then yeah, they were doing some analysis and comparing, like to other classes of gliomas profiled with single-cell RNA-seq. So it's basically how many? We have 2.5 thousand cells. This is H3K27M patients. Then we have some glioma models, and then some gliomas. So okay, I already prepared the data sets, so I put them on a server. So can you please access this URL? So if you click here, right, you go. Okay, I can click. You should you should type it in your command line of the browser. The presentation? Yes, I can. Yeah, I can send the presentation. Right now, it's easier for me to send you the link. I can send you a presentation later after the class. Yeah, just a second. There are a bunch of them, so I just, I wanted to prepare some files. Okay. Okay, so I've sent you the link. You can access it from your email. So basically, the first one which we'll look at is K27M project RSAM, this file, DB file. It's 65 MB, so I'm assuming that you have enough space on your laptop. Okay. Also, I would like you to download PBMC 4K zip file. This is just in case if you have more time, we will analyze 10x dataset as well. So, okay. So the first one is this, and then the second one is this. So you were supposed to download this one, K27M project, and PBMC 4K. 4K.zip, yes. Okay, so the first one which will analyze this is K27M project. So you don't need to do anything with it. So let's see if we have some time at the end. You'll probably need to unzip PBMC4K. You can do it right now if you want. Okay, let me see what I have here. Yeah, so for example, here in my working directory, I have this one, right? And I have basically also PBMC4K. But not necessarily, fine. Let's think about this. If you put your files, let's say, to a directory, let's do the following. I'm suggesting you to do the following. So first step would be create a directory. So for example, if you're on Mac, it will be somehow like users, and then your here you, and let's create UCB single cell. Okay, so that's why we will be on the same page. So here, instead of you, if you're on Mac, so it should be your username. So I can show you what I can do here. Is the following. So this is me, users, my name here, and then I can do either from the terminal. If you can do it, it's okay, or you can go to Finder and create the directory from here. So let's say I can come here, and then, okay, how do I create a directory? Okay, I have no idea how to do it from Finder, but I know how to do it from here. So PCB single cell, right? Mac. So Windows, right? You have Windows. So then you just create, as usual, as you do. It's all the same. I don't know how to do it on Mac, except for from the terminal. And then place your files here inside of this directory. So this is what I have now. I show you the contents of this directory. You will see that I have it. Okay, this is my home, and here I have PCB single cell, and this is the file which we just downloaded. Question? Yes. Not necessarily. This is the way I'm just doing. So you can do it direct from Finder if you know how to do it. I'm not very familiar with Mac. But in the end, yes. You can do it if you know how to do this. This is just for us to be easy. Because usually this step takes a lot of time. People have different operating systems, so that's why I'm suggesting you, if you are on Windows, you do it the way you do it on Windows. On Mac, do it like you do. If you're on terminal like me, you can do it this way. Plus, the addressing is different. For example, backslash or slash in different systems. That's why we need to kind of, if we have, the majority of us have Macs, so then do it this way. Otherwise, if you have Windows and you have problems, you just can tell us and we help you. So just to make sure that you, like when you enter this directory, or you can download it to anywhere to your hard drive and then set the working directory. That's what we will do, but just let's have it coherent across all the people. Is it done? Did you guys do it? Okay, good. So fine, let's open RStudio then. Okay, and let's run the script line by line. So basically, okay, let me probably try to enlarge the font because, okay. Oh, okay, just a second. So, okay, for tidyverse, that's what I'm suggesting to you. This is a small exercise. Let's do a small exercise here. If you don't have this package, so you install it probably the way you install Seurat. So just type install packages on tidyverse. All right. So I should already have it. So please type this command. Install packages tidyverse. Yes. Zoom. Yes. Now you see it. Okay. So if you install it correctly, then you're able to execute this command. Okay, and then you'll see some weird messages. So basically, when you import this package, it also import for. If you see this kind of thing, then it means that it's okay. Do you see this? Good. Anybody else having troubles? So let's make sure that everyone has this, because you might not catch up later on. Okay, if you install this, so let's basically the first step in this analysis would be setting your working directory. So for us, I was suggesting you to do something like this, right? I had something different, but let me change to the one which I just created. So this would be QCP single cell, right? And then that's it. This is my working directory. So you can go, for example, here to Finder and then check where your file is. So here for me, if I look at the path, right, it will be users, my username, PCB single cell, and then the file, right? For Windows, you mean the path? Yeah, I think you should C or D, like it depends on which disk you are. But then, it's, yeah. But I think that even though in Windows you see that the absolute path have backslashes, right? So anyway, in R it should work like this. Okay, so basically, if you did this, you can like easily check that you are in the right directory. Let's say list files. There is a command list file. So basically, in this directory right now, we have only one file, right? This is the one we downloaded. You have it? Yeah. So next, if you are ready, let's load the data set into the memory. So it will take some time. So basically, what happens? You don't need to gunzip the file. So now it's possible to read in the memory in compressed form. Yes.
So if it's an Excel file or like a GC file, what's the difference? Is there any like even like better?
Better, like what would be better?
Let's say you load a data set. I noticed you did three framework GCs, so like a text file, but then I've heard some cases Excel file. Like, does it matter? Data sets?
I think it does not, in general, matter for your work, right? So this is a text. So basically, okay, we can look inside of this file. You will see that this is just tab separated file, but it was saved as txt. So if you have in Excel, but most probably I will assume that you won't. Excel, like, you will not operate with this data sets from Excel most probably. You can have .csv, for example, where your delimiter will be not a tab but a comma, for example. So you can open it both in Excel and, for example, import it to RStudio. That's all fine. So, but if you're working with extremely large data sets, like this one is not large, so it's more or less like medium. But if you have one with, let's say, 700 or like 1,000,000 cells, so then you'll probably need to store it as a sparse matrix, because we just learned yesterday that these matrices are very sparse. So usually they have, let's say, 5 to 10% of the values non-zero. So instead of like saving all the zeros, there are specific formats for storing such matrices where you only memorize the coordinates of non-zero elements, and you have a vector which stores the actual data, which are non-zero. So you don't need to store zeros with so many zeros. This is in this case. So Seurat, I think it can basically inside it has this mechanism of sparse matrices. But here, okay, we can just look at this file. So I can show you. Okay. Here, let's see. So this is only one line. So it's a huge file. So basically here we have gene, and these are I'm assuming cell names, all right? And if we go here, so this is basically, so this is the gene name, A1BG. There's such a gene, right? And all of these things are, most of them are zeros. And here we have some counts, right? So these are not actual counts, but these are TPMs. Remember TPMs from yesterday? So you have a read count from a gene. You divide the read count by the length of the gene, and then you divide by the total sum of such numbers. So here, the numbers basically are non-integers. So they're TPMs, and we know that they sum up to 1 million for each cell. Okay. Oh, yes. How do I do this? Where is it? Oh, this is, yes. From raw reads? So what kind of technology do you have? SmartSig? Okay, yeah, good question. So if you have raw reads, let's say, right? For example, if this is 10x, then you run them through 10x pipeline, which is called Cell Ranger. It will produce you this kind of matrix, right? Or otherwise, if you have something, let's say SmartSig2, you will need to take, because the SmartSig2, how the data looks like is basically each cell is a separate file with reads. Then you will need to take all these files, run like alignment, then quantification, you know, and then combine them into a matrix. There's more steps you need to do. Right? This is your question? Or? Yes, in case if you have SmartSeq2 for 10x, run Cell Ranger. So you have some, let's say, some raw files. There is a command mkfasq, so which will make you FASTQ files. And then the other one, I don't really remember, it's probably like Cell Ranger count. It will produce you the count matrix. Right? So all the steps are implemented there, and you don't need to do. So as output of that pipeline, you will see this kind of matrix. Right? So here, so this is just only one line, you know, like we have several thousand cells here, right? And the data is very sparse. Some of the genes are more expressed, you know, in many cells. This is how biology works. So basically, when we see such zeros, right? Remember what we were talking about yesterday. So when we see such a zero, we don't know whether this is a biological zero or this is a technical zero. So we call it, okay, it might be a dropout. But nobody knows, you know, like people still are debating about what are the zeros in this matrix. So, sorry, what?
It's just too low to detect.
Yes.
We don't know the action out there.
Yes, we don't know. And like, for example, you know, like many papers were suggesting that this is zero, like, for example, technical one. And then there are like a whole bunch of methods which impute these unknown values. So this is imputation. But recently there was a paper which said that, no, you know, guys, so these zeros are biological ones. So they argued a lot about, you know, the fact that this is a biological zero, which we don't know. So probably, you know, in some like 5 years or 10 years, people will answer this question. But for now, people are still arguing. Okay. Yeah, so if we would have 10x matrix, so basically we'll have such a matrix with integer numbers. This will be UMI counts. So don't confound them. So these are TPMs, which were produced from full-length transcript technology, right? And for 10x, we have UMIs, so basically molecules. So here in this case, we're counting reads, which we get from the sequencing machine. In case of 10x, you get this matrix with integer values, and these integer values are molecules. So for example, if you see in 10x, let's say if you see 5 somewhere, right? So this 5 means that you have 5 molecules, right? So it means that you have 5 molecules. And when we look at this number 5, we don't know how many reads were behind the scenes here. Because, for example, this molecule probably was observed with 10 reads, this one with 50 reads, and so forth. If you have all the files from 10x, you can restore this information, you know, but people usually do, you know, don't actually look at this. So they just produce the final counts and use them in downstream analysis. For our case, so this first data set is actual reads, reads from here, because we don't have UMIs in SmartSeq2. So this were reads, and we computed TPM. Okay, so you should understand this distinction. Okay. Okay, good. So this is the file. Do I need to share now this RStudio?
Yeah, you should.
Okay.
Sorry.
This is. What? Yeah. So those of you who have Windows, so did you have any troubles with loading the data? Yes.
It says that it cannot open it.
So should Ian, can you please help, or should I look at it? Okay, so let's do a quick break. Okay. Okay, so for example, we want to look at the first 6 rows, right? There is command head, but because we have so many cells, and basically our matrix is like this. So the rows are genes, okay, and the columns are cells. So that's why, because we have like many cells, we can find out with the command dim. So what it means that we have 4,000 cells, and each has 23,000 genes. Okay, that's what we have now. Okay. Now we have this TPMs. Let's call them directly TPMs because they're not counts. We have TPMs. What we are supposed to do next is to create a Seurat object. So in Seurat, we're dealing with basically there is a central thing, right? An object which is called Seurat. Right, and the very basic command, like the very first command we have to execute, is to create Seurat object. So first of all, the first parameter here is my data. This is the matrix which we imported to RStudio. So here you will see that this function also has some arguments. Like the first argument is primary one. So this is the argument called counts, and we supply my data, so basically the matrix. But it also has some arguments, like for example, minimum cells is 3 and min features is 200. What it means is, remember I talked yesterday about QC step, so quality control. So what it means the following: this matrix is very sparse. If you look at some genes, okay, let's look at some genes. So this is okay. I need to show. So this gene, right here, AARS, it's more or less expressed in a lot of cells, all right? But okay, this one is very good. But there are some other genes like this one, which is very sparse, how would you call it, right? So it means like at least I can see here in a bunch of cells at least only one value which is expressed, 23.8. So most probably this gene is not really interesting because it's only present in a couple of cells out of several thousands. So that's how you would like to eliminate this gene from further analysis, right? So in order to do this, so you specify this argument min cells. So what min cells means that we want to take only those genes which are present in at least 3 cells.
Like some sort of best practice for that?
I think, okay, let's build the fast plot and then we'll talk about these practices, because it depends. It's really, everything is data dependent, because all the data sets are so different. And min features, it means that we are taking only those cells which have at least 200 genes expressed. If you have, for example, 100 genes in that cell, then something is wrong with that cell, right? Because we have 20,000 genes, and if you observe only, let's say 50 genes, something is weird.
The min cells option eliminates genes that are seen in 3 or 4 cells.
Yes. Okay, so now let's see. Oh, okay. Oh, no. So I need to go to this. So, okay. Now we will basically operate with this object glioma, right? So this will be the main variable. Okay, so let's create the object. Did I do it? Because I need to import Seurat. I did not do it. Okay, so
You really need that tidyverse one.
it will appear only like one function from that tidyverse here. So let's keep it.
We'll try to restart all of it.
I mean, it has tidyverse, and there are a lot of packages which are from the same developers which are useful with data manipulations. So it's good to know them. Okay, so I just have created the Glioma object, right? So if you type it like this, you will see that it tells you that this is an object of class Seurat, which has this many features across 4058 samples, right? And let's recall what was the dimensionality of my data.
So we don't need to care
About what? No, this is, okay, let's read this warning. So what it tells, feature names cannot have underscore. So it says that the gene names, so it calls genes features. So it says that gene names should not, cannot have underscore. So some of our genes, they have underscores, and then it replaces it with dashes. No, well, but it might be some confusion sometimes. You know, if you're assuming that your gene has underscore, but inside of Seurat object, it will have a dash. So it might have some issue. Should I? Okay, so if you look at the at this message here, we see that we have 21.8 thousand features, right? And initially we supplied 23.6 thousand features. So the question here is like a small exercise for you guys. Why do we see? Why did we lose some features? Any ideas?
genes that were only in one or two.
Yeah, so this was probably because of this filtering, right? So basically, if we adjust these parameters, we'll see, you know, different number of features. So okay,
That's only because the min cells thing.
yeah, yeah. In order to prove this, let's take, for example, 300.
What is min features?
So this means that we take only those cells which have at least this many genes. They call them features because, you know, it can be used also with, for example, ATAC-seq, or you know, you call features in general.
So the cell has to have at least 200.
Yeah. Now, for example, if you run it for 300, right, you will see that probably your number of features will go down.
It looks like no cells got lost.
Okay, let's see. Now I have only 13,000. See, so basically, if you look at this parameter, I changed to 300, and now I have only 13,000 genes. Okay, fine. Let's do it like this. So we'll have, you know, lighter data set to work with. So it depends. Okay, let's look at the plot. So in order to understand this, you plot the things because you don't know in advance what to do. So initially, what I would do here is when you don't supply these parameters, so it will take everything, and then you will plot the cells, and then based on that plot, you will understand how much to trim. So if we jump to line 29, all right. So what will happen here is the following. Okay. So we'll have these two plots. So the first one is telling us, okay, so number of features. So it means basically, you know, it will tell us each dot, here is a cell, and it tells us how many features we have expressed, how many genes expressed. Okay, because we started to some number, right? Probably that's what we see here. It depends. So we will try to do the same plot when we don't cut anything, right? And we will see how it looks like. There are some outliers, but not extreme ones. And the second plot, it tells us number, you know, like basically the total counts for each cell. And so this is the distinction between TPMs, SmartSeq2 and 10x. In 10x, this number is variable, you know, and remember for TPMs with SmartSeq2, we said that they sum up to 1 million. You know, so that's the distinction. So if we would do like, okay, we'll have some time, I hope we'll have some time at the end. When we'll analyze a 10x data set, we will see more variability here. But here we just have, like 1 point. This is something, I don't know what happens in those cells, some weird things. But most of them lie here at 1 million. So this means that they sum up to 1 million, and there's almost no variability. So believe me, there are like many more cells than in this outlier section. So, okay, let me try to plot the things when we don't have these arguments, right? So what I will do, I will just, for example, comment it out. Okay. And then when we plot these things, it doesn't change too much. Yeah, so basically most of the counts are here, right? This, between basically 1 million. It's all of them 1 million plus minus some, some epsilon, which is just some, error, small error. So in this case, all the cells look really good, I think. But sometimes, basically, when you look at this plot, this is actually a violent plot because there are so many cells. We don't see the violent things, but it's there. If you look carefully to what you have, you will see a violent plot. And basically, this distribution will tell you, okay, if there will be a lot of cells which have, for example, less than, let's say, 2,000 genes, then probably or like 1,000 genes, probably you just, remove them from the data set. So very, very extreme values. So you should look at this plot and say, okay, so for example, if a cell has too many features expressed, right? This might suggest that this is a doublet if it's 10x. In SmartSeq2, we don't have this thing. Okay, so that's why it's good that we have we retain all the cells. For 10x, it might be a doublet. Or for example, if it's too low, or the number of like the total count is too low, then we might need to consider, removing the cell, because most probably, if the count is too low, then this is just an ambient RNA there, and there is no cell in the droplet. Okay, again, I'm just repeating. This is SmartSeq2 dataset. Yes. It's giving you an error, this violent plot?
Yeah.
Oh, I don't know what you should do. Let's see. So, yeah, ideally. Okay, I think. How to check the version of Seurat? Oh, here?
Yeah.
Oh, yeah, I see. Okay. So if you look at Seurat, okay. .0. Yeah. Yeah, if you're okay with installing 3.0, it would be great. Yeah, I think 3.1, I don't know, like maybe it's not that yet stable, So the 3.0 is already used many times. So they changed. So in Seurat 2.0, they had, some different syntax also, like for what we have now with different minor versions. So what I assume might be wrong is probably the names of the features. So here, instead of N feature RNA, they might have some different names of this fields in that object. Because previously they had N genes and N UMIs. That's you can try this thing. If you see, I have this in comments: N genes and N UMIs. Okay. Okay, so now let's see. There's a function feature scatter, which allows us to do basically different plots. So here we can take any characteristics of each cell. Let's say we want to plot, you know, like number, let's say here in this plot, what we see here is on the x-axis is the total count of, you know, of genes, and then like how many genes are expressed. Sorry, not the how many genes, so the total count, the total read count. Okay, because we have TPMs, we all the time have something which is 1 million, right? And here is how many genes are expressed. So what this plot can tell us is, for example, if something is wrong with some cells also, you know, it can be used for some kind of diagnostics. Like, for example, you might, if you plot here, you know, the total read count versus the number of genes which are expressed. Okay, if you see that some values are skewed, like for example, in this case, right? You see that, okay, the total content of the cell is not high, you know, but there are many genes expressed. So it means that on average, each gene is expressed less, and probably you also, from some, you know, following some considerations, you'd like to remove it. So this is just to, you know, to make sure also that our data is okay. Oh, you guys want 300? Let's work with it. Yeah, okay. I see that you probably you have different picture. Okay. Yeah, well, this is feature scatter, right? Oh, that's what you see, right? Okay. Yeah, so what happened here is probably because we removed some, you know, 300 cells, probably we see now something which is basically, it means that, you know, we removed some cells which for some reason had this TPMs greater than 1 million. I don't know, it might be some numerical instability when you compute this values, but fine, this is TPM, so this is a bit different. You like in TPMs you should have everything 1 million, but for some reason I don't know. Like we have this data, you know, this data we should not trust it because like who knows like who did this data set? We just took it from the internet. So they, yeah, you know, like for example, it might be something when I can tell you what can happen here. So for example, you had a cell, right, and then you computed the TPMs. You have, you know, fractional numbers, and then because you cannot probably, okay, you can, but usually you have to trim the number of you know, significant digits after the period, right? So that's why basically when you take, for example, the two most significant ones, right, what will happen that you will lose from the total sum of 1 million. So that's why we see this cells here, right? This is the way how we do it. We do it to be 1 million correctly. This is TPM. Yeah, so when we do, okay, just, you know, in parallel, it would be great if we do a data set with 10x, but let's do one by one. In 10x, we will see variability because this would be read counts. Here we normalize them, we do it all 1 million, right?
It came in normalized, but then it got a little unnormalized, and there's a handful of cells that are only 1,000. My guess is those are the dropped out. Those are genes, cells that had a lot of it.
No, but it should have 1 million, and probably there was some, I don't know, something went wrong with some of the cells there, because 800,000 is, it cannot be explained by, you know, numerical things. Numerical is something which is closer to here, and this one is something weird. Okay, we can, for example, also think, okay, maybe we should remove these cells, for example, right? It depends, but in general, you know, like if your data set is okay, if your data set is good enough, then probably you will not want to remove anything, because if you will cut a lot, right, you will remove, for example, a lot of cells, and then let's say the power of your analysis will be limited, right? So for example, differential expression between clusters, you need to have more cells to have more power to distinguish between different marker genes. So that's why you some cells might look suspicious, but you still might want to keep them. I think you know, like there's probably not a, you know, as single, you know, like gold standard for this, because it's all data set dependent. Like for example, in one case you might want to remove a lot of things because, I don't know, probably someone in the lab messed up something, and then you will want to, you know, remove a lot of things from your data set. In some other cases, when you know that everything is of high quality, then just retain everything. Okay, there is one more thing what we can do. Well, you know, like for example, in the beginning here we were loading 300, you know, but 300 we saw that it's too much, so we are basically removing a lot of genes, right? Initially, we might set it to everything, like to a small number. Let's say 3 instead of 303, right? We had 3 in the beginning. But then if you, like after, let's say we look at those plots and we understand how much to cut, right? Then there is another command which is useful for QC. So it's subset. So for example, if we want to take a subset of cells, you know, for which basically number of features, so basically number of expressed genes is greater than 500, but it's less than 8,000, right? So this is the command, how to do it. So let's all execute it and have smaller matrix. We do not have a large one. So after we execute this, indeed we have how many? We have already a smaller number of cells. So basically we removed like 200 cells. Okay, 150, something like that. So this subset, it means that take only those cells for which we have this condition true. And if we, you know, basically, if we put some restrictions on the cells, right, it means that in the new data set, after we are subsetting it, we will see less cells. That's what we see here, this many cells. So samples means cells. So feature is a gene and sample is a cell. Okay. Are we all on the same line here? Okay. Let's. It's good if we will have some time to go through for a 10x data set, you know, see what would be the difference. So next, you know, like after.
Those QC numbers are different.
Yes.
In feature because they're all like 20,000.
Yes. So we'll look at the plot and then we'll decide. Okay. Yeah. That's what you usually do. So now basically what we have at this stage, right? We downloaded the data set, right? Or for example, you get it from a lab, or you sequence your data, right? And the technician comes to you and brings you the data. Now you load it here, let's say in RStudio, and then you do some QC steps. Next, what happens? You need to pre-process the data. So here it's good that, for example, SmartSeq 2, they are already, you know, scaled to have 1 million. In case of 10x, each cell will have different number of UMIs, right? So the first step would be what is basically to scale data, right? So we'll need to basically scale the data. So let's run these two things. Basically, we say that we take all the genes from this object, you know, and we use all these genes to scale them.
Can you say scale for this stuff? Does that mean what?
Okay, scale is supposed to be, okay, I think in this version of Seurat in 3.0, when you do scale data, they probably also, so basically what they do is usually it's supposed to be having the same sum for all the cells,
Oh,
right? For us, we don't have like much effect here. Yeah, for 10x it will change your, right?
So scaling is just do it that way. No judgment.
Yeah.
Unless you got a reason to scale on that subset.
Do all the things, yeah. So okay, let's also normalize. So I'm suggesting you to skip some of the lines here, you know, because it will take some time to explain. So basically, I was showing in this line how scaling or how normalizing works, but basically, let's go to the line 55. In the line 55, now there is a command for normalizing data. Normalizing is taking the log. Okay, it's not actually log, but one log of 1 plus x, because we have a lot of cells having genes with expression 0, and that's why you cannot take log of 0. So that's why you have to add 1 first and then take the log. 1. So 1 plus x. So basically, you run this function normalize data. What it will do, it will okay. There is also you know, something like scale factor. So now all the cells. Okay, let's have 10,000. Let's have them to sum up to 10,000. I think this is what people do in single cell. they scale them down to 10,000. So now what happens? You add, for example, this is your expression. You add 1 plus x, and take log. Okay. So for example, if you have 0, you cannot take log of 0 because this is infinity, right? So you cannot compute it. So that's why you have to add 1, because log of 1 is 0. Okay. Okay, good question. So, okay, let's return back a bit to the workflow. The very first command when we create Seurat object, right? So here, these two parameters, 300 and 200, you don't know how much to set in advance. So I'm suggesting do not set them at all. So you will take all the data, you draw the violin plot, you see, for example, okay, how many outliers you have and where you should trim your data set, right? So like how many cells to take. And then when you are decided with this, you know, like how to, you know, take a subset. You'll need to take a subset of this. And then there is this command subset. This 508,000, I just, you know, took it randomly just to have a smaller data set now. But in the case when you analyze a data set, you should look at those violin plots. For, you know, for SmartSeq2, everything is kind of normalized. So that's why we didn't see many, you know, outliers. For 10x, we'll see, hopefully we'll have time for 10x. If not, then this will be your homework. Like you can take 10x and just literally run the same script, you know. So you will see that, you know, you'll need to cut it somewhere. Like, let's say some cells will have a small number of genes or very large number of genes. You just want to remove them from the analysis. So now let me show you something. So basically this object Guillaume, right? So if you type it like this, it will show you that this is a Seurat object. Now, if you want to somehow access basically what, let's say the initial counts, right? Or initial TPMs, the raw matrix. So this is what you do. You call glioma and then add a space, $RNA, add counts. All right, so this will give you the very matrix which we downloaded, and which we can see here. This is the same matrix here. The command, okay, the command is this one. So it will be basically something. A space, $, RNA, add. So inside of this object, there is a path inside of this object because it has a lot of things. So basically, when we do all the analysis, nothing is lost, but it's being stored inside of this object. When we do all kind of manipulations, it's all. Oh, what is the meaning of this things? So okay, these are okay. Okay, fine. So let me tell you. So basically, this Seurat object, it's, if you are familiar with R a little bit, you'll know that there are some basic types, like list. You have arrays or vectors, right? You have data frames. So these are basic things, and on top of these things, R has also so-called S4 objects. So if you know, okay, if you don't know, you can read, but I don't know if you understand much. It's a weird, tricky system of objects. So basically, you combine from smaller, like from more, from more elementary things, you combine complicated objects. So this Seurat is a complicated object which stores a lot of things. So it stores counts. So this add is used to to access the fields, or access the fields of this complicated objects. This dollar is usually accessed to to take some values from for example, from a list. So that's why it's a weird, it's crazy, it's a bit crazy thing. What you should get used to. So it's just this object is a hierarchical structure of many simpler objects. That's why you you indicate first at and then dollar and then at. Huh? To what we downloaded and loaded inside of RStudio. But here it also has, for example, data. So this data it stores already normalized values. So log transformed. Okay. Yeah, so like it has, Seurat has a lot of features. So this is one of them. For example, if you want to take, let's say this is expression of one gene, right? This is the way how you do it. Or no, of one cell. This is one cell. Just execute this command, right? Basically, line 64. If you execute this, you will see that the expression here is just literally one cell. Oh, which one? Which command? Data or counts? When you do this? When you do this? Okay, so let's go to line 73. Okay, so basically here on this line, I will execute command find variable features. So the idea here is that in single cell, right? So we have basically, as in all RNA-seq experiments, we have a lot of genes. Because of the fact, again, that some genes are not expressed, or some genes might be, for example, constant across different cell types, so they're not interesting, right? If they, like for example, if you look at one gene and it has the same expression in all the cell types, so you don't gain any, knowledge of analyzing this gene. So what happens? You probably want to take only the most informative genes, right? So what would be the most informative gene? Is the one which has the highest variance across the cells, right? So this command here on the line 73, it tells us that we take the object glioma and we select only top 2,000 most variable or highly variable genes, right? So it's a kind of long command, and all the commands in Seurat, they have a lot of parameters. So basically, okay, let's try to look at it. Find variable features, right? So you can type, a question mark, and then you can type the name of this function, and then you have the description, right? Like you can use this functionality in RStudio to read the description of the functions. So it identifies features that are outliers on a mean variability plot. So basically, it uses some, so it looks at basically some, calculates the variance to mean ratio. So if you look at here in the comments, I specified this kind of things here, right? So it looks, at the variance of a gene divided by its mean, right? Because what we usually assume that in RNA-seq, if a gene is highly expressed, then it's also highly variable. So, when we select the highly variable features or genes, we also need to account for this fact, So that's why we select the genes not based on their variance, but based on, variance divided by their mean. So mean and variance are coupled in a sense in RNA-seq.
Sorry, that's done by default when you find variance.
Yes. Yes, I think this VST method, that's what it is. So it's by default. So here, VST. So that's the description. It first fits a line to the relationship of log variance and log mean using local polynomial regression loss. Then it standardizes the features values using the observed mean and expected variance. And then feature variance is then calculated on the standardized values after clipping to a maximum. So this thing is, I think, is by default. Okay. So here, number of features, I actually don't know what's the default here. But usually what I have seen people use, let's say, a couple of thousands of genes. So not 5 is already too much. So 1,000-2,000 genes is okay. All right, you will probably capture most of your biological signal with this many genes. Okay. Yeah, that's like after we read this, the manual, right, for this function, you actually see that it first calculates gene variances, right? Says, so you can see it here, feature variance is then calculated on the standardized values after clipping to a maximum, right? And then it says what calculating feature variances of standardized and clipped values. So it does some, some kind of transformation to find out which genes are most interesting ones, right? So for example, we can take, let's say, top 10 genes, right? So this is command on the line 74. To give us top 10 variable genes. So these are these genes, CD74, right, and all the other ones. You can have, for example, like the top, if you want to look at top 100, just change here to 100. If you want to look at all of them, just use this function variable features. All right. So it would be basically variable features, and this is your object, and it gives you all 2000. Okay, now also in Seurat we have this kind of plot, which is variable feature plot, all right? So let's see. Okay. When we execute the lines 75 through 77, what do we see? Okay, I cannot enlarge the plot. No way, because I have it here. Right, so basically here what we have, this is average expression, and then this is variance, right? And we selected only the genes which are highly variable, right, which have higher variance. So these red dots, we have count of them 2000, and then 11.5 thousand are non-variable ones. So now usually what we do for, let's say for some other downstream analysis, we're only dealing with the variable genes. Again, so we're like with all the steps, with each step we are removing some things which we will not use. So we are basically using here only 2000 genes, and these are the top 10 ones, right, which we printed initially. So they're like really, like in a sense outliers. I think it's too much variance for such genes. Okay.
And we have to keep in mind mean expression as well, just because ones that have higher expression will vary more.
Mm-hmm. Yeah. Not necessarily, but it's.
Okay. Just like if it's a larger number, you have to scale it by the average.
Yeah, that's why they take so big.
And just suppose, for example, you have, like you observe, let's say, a gene which has, like, let's say, in most of the cells, the mean value is like read count is 2, right? If it's 2, okay, probably, if it's, if the genes, for example, if you're familiar with, let's say, some statistics, you would think that, okay, maybe the read count is a Poisson random variable, and then it's so, then, the variance of Poisson is coupled with the mean, and it's actually equal to the mean. So that's why if you take, for example, a gene for which mean read count is 2, then the variance would be 2, right? And then if you take one gene which has mean 100, then the variance will be 100. So that's why, here we also need to look at the mean. Okay, next, let's recall what is PCA, principal component analysis. So in principal component analysis, if you remember the slides from the yesterday class, is what we are doing, we are doing a linear transformation so that now our axis will point to the directions of the highest variability, right? So because now we'll be using here, like, let's look at the line 81. So here in this line, we are using only variable features, okay? So basically now our data set, is kind of, okay, it has 13,000 features, but when we are computing PCA, or principal component analysis, right, we'll use only 2,000 variable genes. That's the usual thing, in all this analysis. So we reduce, and then, we with PCA, we get even less dimensions. So if you execute this command, okay, what happens is that now we'll get the,
In this case, we're refinding variable features. No, we're not. So if we wanted to try with a different number of features, we'd have to rerun find variable features.
Yeah.
Whatever you've done previously. That's it.
Yeah, you just select a different number of variable features, for example. But usually 2000 is okay. Like, for example, if you play, like, what can happen if you take less features. it might happen that when you will look at your clusters, they might not be well separated, you know. So that's like sometimes in order for some clusters or some cell types to be well separated, you need to consider more dimensions. So maybe in some of the dimensions, they will be well separated. If you remove those genes, you definitely, you know, you are smoothing the frontier between two cell types. So here, let's look at the output. So you should see something like red.
Excuse me, for doing PCA, is it just a time saver that you're only using variable features? What if you just use all of them? It would take a lot longer. But
it might take longer, but you know, well, one thing is then okay, you're losing some information. My assumption is that, and I think that you can really also check this, that if you do it on all features, you won't lose much information, because with taking 2,000 most variable genes, you know, you're selecting most of the variants. Now, what happens with all these variants? Because anyway, it might save you some time. Yes, I agree. But for this small data set, it will not be, you know, big save.
yeah. So it seems like you might want to err on the greater side of features, of variable features to use.
Mm-hmm. Yeah. So now when we look at the, okay, it comes up as a warning here because it's like red color. Do you guys get this PC1? So basically this PC1, this is what, this is the main direction, right? The principal component one. And we see there are some genes which are like positive and negative. What it means that along this direction, you know, some genes basically will have, you know, will be kind of contributing positively, and some of them will be contributing negatively to this direction. Okay, so principal component is just, you know, a linear combination of these genes. So these genes, they contribute, because if you look at all 2,000 genes, right, it's difficult to understand what's going on in this many dimensions. Now, when you reduce the number of dimensions to, for example, here in Seurat, I think the default is 50, but you can take any other number, right? So basically, now you're like projecting your data, you know, to some space, which is some subspace, which is not necessarily, you know, orthogonal to some of the genes. So this means that as a result, your projections to that subspace will, you know, will be basically some linear combination of the genes.
And if you do linear combination, the negative genes have negative coefficients, and the positive genes have positive.
Yes, with that direction. Yes.
Yeah.
Okay. So this is you know, we can read these genes, that is fine, but okay. Here we also take, for example, number of features, 20, right? We can basically print the same red warning message here with this command on line 84. Okay. So remember, I was telling you that Seurat object is kind of, you know, it has complex structure. So what it means that after we run PCA, it does not, you know, it basically stores the result of this PCA analysis inside of itself. So what it means means the following: if you take this one, all right, if you execute command on the line 88, you will see that here it will tell us what this is, a dimensionality reduction with what I was telling you. By default, it takes 50 principal components, right? And okay, it has some other messages here. This is it a warning or an error? Because this happens when it's doing SVD, singular value decomposition, inside of PCA function. So okay, did you follow the same steps with us, having the same number of cells? Sometimes it happens, you know, like it happened to me frequently, but I think it was something wrong with the data usually. Do you also have? No. Is anybody else having? Oh, because you have to run. You have to fast run this 81 line 81. You did not run it, so you need to run this. So after you run this command, it places the results of your PCA in this PCA slot. Okay. Yeah, now you should have this PCA, and basically what I'm thinking that, yeah, it has this PCA object. You see, it has some other things, like for example, cell embeddings, feature loadings, and then feature loadings projected. So for example, this cell embeddings, it will be what? It will be basically the coordinates of the cells in the new coordinates of the cells, right? So we can do something like this. Let's say we can look at something like this. So here, when I execute this kind of command, right, what happens is that I'm taking cell embeddings. What is embedding? Embedding is basically, like what happens, our data set doesn't change. What changes is the coordinates of the cells, right? Because we don't do anything with the cells, we do with the coordinates. And then we are trimming the coordinates based on like how informative they are. Right. So here in PCA, usually the first coordinate is the most informative, the second one. So we are ordering them by the amount of information they explain. Right. So here, for example, we can take this PC1 and PC2. This will be, the two most informative directions. And these two numbers in cell embeddings will be the coordinates of the cells. Like, for example, this would be this cell. And this would be the coordinates of the cell in the first two principal components. Okay, so this is cell embeddings, all right. And there is also, we can look at, for example, feature, what is feature loadings? Feature loadings, right? So this feature loadings will basically indicate us how much, okay, wait. Okay, let me remove it. So this feature loadings will tell us how much each gene is contributing to each of the principal components. So basically, if you think about this PCA, right? If you, for example, if you have one gene which is, which has a lot of variance, right? And after doing PCA, hopefully that, the first PCA will have very large influence of that variable gene. And then this means that the loading of this gene in the principal component will be very high. Okay. Okay. Oh, I already have this line here, line 92. So it shows us the embeddings. Okay. So now I was telling you, right, that, for example, the first principal component explains a lot of variance, right? So let's check this kind of thing. So when we execute line 96, right, we are checking the standard deviation. So now look at this, we have 50 numbers, right? So there are 50 numbers here. And the first one, it has, standard deviation of 8. So this means we take all the best coordinates of all the cells, right? And then it will have this much standard deviation. And then the last, so they're basically in decreasing order. So you will see that the last one is explaining even less. But this is 50, right? So so now, like if we have 20,000 genes, right? So PCA, like it ideally will compute you 20,000 principal components, which will give you the total variance of your original data set. Out of this 20,000, the very last 20,000, like the very last couple of, or not couple, like even thousands, they will have such a small variance so that you will, just ignore it. So because you see, like if you plot these numbers, right? let's see if we can plot it. Oh no, this is feature loadings. This is something different. I want to plot this. Okay, so you will see, this is kind of exponential decay. I don't know if it's exponential, but it looks like, so it basically starts with 8, and then it approaches 0, and if we take not 50, but if we compute 20,000 principal components, which will correspond to the dimensionality of our data set, it will be like very close to 0. So some genes will be, almost constant, almost 0. Okay. So there are multiple functions in Seurat which can help you visualize your PCA analysis, right? So for example, we can visualize dimensionality loadings here, right? So basically, these are the scores of the genes in each of the principal components, right? So remember, we were looking at feature loadings. So basically, this is just a nice plot, it's okay, you can use it. I don't know how crucial it is in your analysis, but this is just a way to visualize which genes contribute to which principal components, right? So these are the genes, which have coefficient minus 0.009, and then some other ones. Okay.
And these are the top genes that contribute to that.
Yes. So here we're only looking at 2, right? You can take, for example, you can change this parameter to 10, right? And then if you run this command, you will see what you will see. You will see something weird. Like you will have 10. Why is it taking so much time? Okay, well, let's change this to something else. Okay, it's frozen now. So this red dot, it shows you that our CD is still, like, R is computing. Okay, so let's use not 10, but 5. I don't know why, or like 4. I think it's too much for it. Now, okay. So these are the top 4 principal components. Okay. Next, we can do a kind of heat map plot, right? So this heat map plot is here in this heat map plot. We took only principal component 1, right? And when we run this function, we indicate that, okay, let's take top 500 cells, with the largest score along this principal component 1, right? And let's take, I think it takes 20 or something genes, and visualizes, basically the score of, it visualizes the cells. So basically it's sorting here are the genes, and here are the cells, right? And then in this 500 cells, it takes you, like for example, these genes are, the colors basically indicate, like the contribution of this, or like basically the, the value of this principal component in these genes and in these cells. Okay, so if we look at this yellow one, I think these genes, they are basically positive in these cells, like in half of the cells. And in the other half of the cells here, these genes will be positive and this will be negative. Yeah, so I wrote you here some comments. So both cells and genes are sorted by their principal component scores, right? And basically, it allows for a nice visualization of sources of heterogeneity in the data set. So we can do this the same, for example, for the first 6 principal components, right? And identify here, right? Sources of heterogeneity. I think, like for example, the first 3 component is kind of more or less clearer picture, but start like for example the 4th one is kind of more or less blurry. And then 5th one is kind of looking good, and then 6th is something similar. So there are functions which allow us to, to take, for example, top cells based on their, principal component scores. So this would be top cells, right? So they usually have some, some coordinates. So for example, if usually I think it should be the top self along the principal component 1, all right? If you take, for example, dimension 2, it will give you, top cells based on the principal component 2. Yeah. And then here I already showed you the embeddings, right? There is another function which allows you to get the embeddings. So for example, for a cell, if you are interested in looking at one particular cell, then using function embeddings, you can take, all 50 coordinates of the cell, right?
When would you want to use these embeddings?
We'll use embeddings in a couple of minutes when we will visualize the PCA plot. Okay. Like sometimes probably an idea might be, okay, just to identify maybe some directions which are like some principal components which are associated with some phenotype or something. This might be the case. So you might use some of them. For visualization, we're using only 2. Right, because maybe some like source of variance which is affecting your phenotype, it might be, for example, not in PC1, but PC4. So, okay. Okay. So there are all these functions, like top features, it gives you, like top principal component genes, right? So these are the genes from principal component 1, and this all this kind of functions, they have numbered dim, like argument dim, right? Which is dimension to use. Like if you want top features for principal component 2, this will be already the other genes, right? You can, for example, specify several. For example, top 3, right? Oh, no, you cannot do it. Okay, fine. It's only one by one. So for example, 50. And the very last one, you have these genes. So now your question might be, so, okay, fine. We have the, principal components. Now, what would be the number of components we should choose? Like how many PCs we should take, right? For this kind of cases, there are different procedures. Like for example, there is a Jack-Straw procedure. It's basically it finds the significant principal components. So because it's kind of confusing, I think, and it will take us some time. So let's not run it. But another like very simple way of looking at this would be elbow plot. Let me. We don't have it here, but I think we can just easily do it. Elbow plot. All right. Just type something like this. So this elbow plot, it will show us. I mean, basically, I already had the elbow plot. Why it doesn't change? What happened? Okay, let's look at this elbow plot. So this plots the standard deviations. Okay. Oh, I'm so sorry. I need to rerun all the steps. Okay. And probably if you repeated this command with me, probably you also will need to run. Okay, let's spend a couple of minutes. So we need to reach this line. But okay, so bad. I'm telling you what happened. I needed to execute this command, elbow plot, but you know, most of the commands it have the syntax as the following one, as this one. But this command, it turned out that it's not. So that's why we overrode the value of the home object. Yeah, because you know I was okay. I was just following the usual syntax of Seurat. So please let's execute briefly from line 18. Yeah, that's so bad. Okay, it should not take much time because but please don't run the Jackstraw. Jackstraw will take some time. Yeah, up to line 126. So please don't go to line 128. Okay, have some errors. Loadings. Okay, in general, now I think it should be fine. So if I write elbow plot, yeah, that's what we should get. So please don't write just, I would suggest you, for example, to comment it out like this. It's another way of looking at, you know, at how many principal components to select. So here, look, actually, you know, we already saw, I did a plot with standard deviation. So that was nothing else than elbow plot. So elbow plot is what basically here on x-axis we have principal components, and by default elbow plot considers top 20, right? We can change it to some other number, like up to 50, because we have computed only 50. So here number of dimensions we can use, for example, 50, right? And look at all the 50. If you probably recognize this pattern because we already visualized it, but with standard plot function. So elbow plot, this is the, you know, it produces a more beautiful plot, right? Which basically tells you the standard deviation of each principal component. And usually, okay, probably, I think here, okay, maybe like something like 20 would be enough, right? Because here you will see that the variance is just almost constant, right? So that's why here in this, based on this plot, you probably will say, okay, maybe 20 is enough. But this is without any like statistical significance. If you would like to do more rigorous tests, you just do Jackstraw procedure. But let's keep it for now and just, for example, use this simple procedure as, you know, here you just decide on your own where to cut, you know, like how many principal components to take. Yeah, I think so. That's usually what I do. It is? I don't know, something here. That's what I think. Okay. Yeah, that's the name of the plot, by the way.
You didn't know anything about the status set. Where would you put
I would pick like something like 22. Something like this. Yeah, I think so. Because still we have a lot of variance in this one, right? 10 would not be enough, maybe for, but who knows? Okay, let's now after we did so. Hope that all of you reached to this to this class. So next thing would be, okay, if it's enough or not, you know, is we can decide by looking at some downstream analysis. Downstream would be what? Finding clusters. So if you take a couple of principal components, you will see that you might repeat this at home, you know, like as a practice. But you will see that if we take, for example, not, let's say, not a sufficient number of principal components, we will not be able to distinguish clusters, because maybe some of the clusters, they, they are like, basically, they are different only in one principal component. Then if you don't take this principal component, they will be just, in one place. You will not be able to distinguish the cell types. Okay. Yeah,
Why
it's
couldn't
better,
we
but
just do all 50 then?
what if we take all 50?
Yeah, like why couldn't we just?
Yeah, you can, but I think it depends on, probably on some trade-off with, like, let's say, speed, like how much you need to, like, if you have a huge data set, maybe let's try to take a smaller one. If you see that you have some troubles with your cell types, or with some other things, okay, you might increase number of principal components, because you might lose some information by disregarding some of them. So that's why I'm suggesting to go to line 138, and let's try to run. So remember, yesterday I was showing some, clustering, and there was a graph, and I was saying that this is how clustering is done usually in a single cell. So this is exactly this function, find neighbors, is basically, first of all, it basically builds the graph, right? And then the other one, here, find clusters, it identifies, cell types, right, in this data set. So if we read the output of this command, so we see that number of communities is 3. So it found 3 clusters. Okay. So basically now, if you want to look at the, identities, so we call identities, these are the clusters where this cell is from, right? So for example, if we look at idents lemma, we see that it has levels 0, 1, and 2. So these are the clusters, right? And here we can visualize like which cell belongs to which cluster. And okay, most of them are 0 probably. So we can look at the distribution of cells by clusters. So I would assume that we need to run command table. So this line tells us that we have most of the cells coming from cluster 0, and then 226 from 1 and 85 from 2. Okay. So we can do many things here, but I'm suggesting to you that we can, for example, do visualization. Let's try to visualize the data. So we can do, for example, t-SNE or UMAP. These are two kind of similar techniques. So that's why let's go directly to line 161. There are a bunch of commands here which you can read afterwards. Like there are just some helper functions to look at average gene expression. To which cells have number of genes less or at least this number. So that's why I would like to skip this thing, and let's go directly to 161 here. So this function, now it will compute the t-SNE transformation. So t-SNE, let's recall what is t-SNE. t-SNE, this is dimensionality reduction technique, which is nonlinear. And so this means that if it's nonlinear, the directions or the coordinates of each cell in t-SNE is kind of, it doesn't have any interpretation. Okay, but we can now visualize t-SNE plot. So hopefully you already run this command. And so when we visualize, that's what we see. Now we can basically look at our data. Okay. No, it's not default. I think, I don't know why I use it here. I wrote 8, yeah. Okay, let's use 10 or like 20. Let's try to use 20. And we can check how the picture will change, right? So let's try to see 20. But with 20, it already, it takes some more time. Okay. No, okay, it's done. Now what happens? Basically the same thing, right? So we took 8 or we took 20, we see the same picture. Okay. So for example, using this function, not this one, okay, we can also look, for example, at, PCA plot. So if you can repeat the same as here on the line 169, right? So this is a way how to visualize PCA. So let's run this. So this is how your PCA is looking, right? It's basically also 3 clusters, right? But okay, at least we know that we can somehow interpret this, right? We know that PC1, it consists of some genes, okay, which contribute to it, and PC2 also. So we can, like, probably if we analyze the set of genes which are most influential in those directions, we can, have some biological conclusions. So PCA in this sense is probably, better to use. But the thing is that in PCA, like, it's really, difficult sometimes to basically, separate cell types. So by default, it should take some, okay, it takes 2 coordinates all the time because we visualize 2 coordinates. But I'm wondering if we can choose different dimensions. So for example, here, by default, it takes principal component 1 and 2. And if you take, for example, 3 and 5, right? This is what, oh, okay, this is what happens in 3 and 5. Look at it, it looks really great. So now cell type 1 is mixed and cell type 2 is basically, they're all mixed. So if you're looking at cell principal component 3 and 5, right, now you cannot distinguish the cell type in PCA. Right, so that's why it's good to take, for example, the first. But sometimes even the first 2 cannot provide you a clear, separation. So that's why we use t-SNE, which kind of more advanced, and it can give us this picture. So okay, we can do experiment 10 versus 11, let's say. Wow. So there are some directions where, like where all the cell types probably have some common biology, right? Like this principal component 11. So they're aligned along this one. Although some cells are different, right? There is probably some subclusters of cells which have something in common. What happens if we go to, I don't know, 49 and 50? This will be, okay. This will be just all the same. Right, so it means what? If you visualize your data set and you are not accounting for the variance, right, for the variability, so it means that if you take these components 49 and 50, you don't see anything and you will not be able to distinguish cell types, okay? And the first 2 ones is really nice. Okay. So in t-SNE, by default, I think t-SNE is basically it's computing only 2 coordinates, but it's enough, like we look at t-SNE and we now see something like this, but it doesn't really mean that this is a real shape of this data set in 20,000 dimensional steps. Moreover, if you rerun t-SNE, because t-SNE is kind of a stochastic procedure, if you rerun t-SNE one more time, you probably will see some different picture. So here, why I use seed_use_23 is that, I just wanted for us to have the same replicability of this result. So this means that you should have the same picture as me. Now, if I change, for example, this to 26, right, and run this t-SNE, I'll see something different. Okay. See, it's already, okay, maybe it's somehow, reflected in some directions, but it's essentially it's the same, but it looks a bit different. So it has some kind of stochasticity inside. So to avoid the stochasticity, we're using this parameter seed use. This is just some fixed number, which instead of drawing it from some as a random number, it will use this fixed number so that when we do all of us, we have the same result. Okay. Now, one of the questions which you might have here is the following. So, okay, we now cluster these cells. So we have 0, 1, and 2. So what are the cell types? So how to understand which cell types do we have here? So this is a kind of, tricky question, because you should have some prior knowledge about what data you're working with. So if you know that this was, what was this glioma, right? So you should expect, I don't know what cell types you should expect there. So probably a biologist should know, I don't know, but a biologist who is working with this kind of data, so biologists should know which cell types to expect. Probably if this is not enough, for example, we know that, there might be like, let's say, 5 cell types. Why are you showing me only 3? So what happens is that there is some parameter in Seurat, like, okay, if we go back to find clusters, if you go to find clusters function, so there is a parameter resolution, right? So this resolution basically controls the number of clusters. So I'm not sure how it will behave now, but let's try to set it to, let's say, 0.5. If we set it to 0.5, now we see already that we have 13 communities. So what it means that, we change the parameter of how the clusters are, are created in this data set. So now we have 13 clusters. And probably like this kind of things will be, separate clusters. So now let's rerun these steps. Like, okay, we ran find clusters, and then let's run t-SNE again.
How do I decide the number of things?
That's what I'm saying. You should know somehow in advance. Like retina cells, stasia cell types.
But like, for example, if I know, so if I know, for example, I have 10 different cell types, how, like, the number I added is 0.5, so.
Oh, yeah, I understand now your question. Yes, this number, they, unfortunately, they don't have, number of clusters because they're using this parameter in the clustering algorithm. And based on this parameter, it will give you some clusters. So probably you will need to, to do some kind of dichotomy search. So you take, for example, a smallest value, you see how many clusters you have. You take a large value, and then somehow, you just refine your search and find the best. I think it's just because of the way how the clustering algorithm works. So it just, because, it might select, like which cells to consider as being in the same clusters based on some distance or some threshold. So this might be some threshold, which, like k-means, in k-means, you can set this number of clusters in advance, right? But here might be, so many issues. For example, some cell types might be, very rare. And then what happens is that some clustering algorithms might not catch it as a separate cluster. It might be, for example, collapse with some other one. So it's a whole art, I would say. Yeah, what? Yeah, so now t-SNE plot is 13 clusters. Now it looks crazy. But at least like here, this is like this one is okay. It's a separate one. This one, different one. This one. It's more or less nice, but still, as far as I can see it here on my laptop, it's kind of weird. Maybe it's a lot. Maybe this number is a lot. I don't know how many clusters are in glioma cells. So, but for example, assume that you think that there are 13 clusters, and then what would be the next step is you just need to label the clusters, right? In order to label the clusters, again, you should know some, some information in advance, like what would be the marker genes. Okay, so because some cell types, they have, like a different set of signature signature genes or marker genes. That's why we need to run this function, find all markers. All right. So I do not advise you to run now this function find all markers because we have 13 clusters. It will take a lot of time. So that's why let's go back and change back this parameter to 0, 0, 1, so that we have a small number of clusters. Okay. Because the more clusters you have, the more time you will need to find differentially expressed genes. This step is very time consuming. Okay. This is t-SNE plot. This is our PCA plot. And then let's run this command. So find all markers, because now we have, again, back have 3 clusters.
It will still take time.
Yeah, some minutes. So we have, okay, let's have some short break. So inside this function find all markers, so basically it also has a bunch of differentially expressed expression tests. So you can use, I think, this function, but okay, we should wait when it finishes. So, or just Google it. Okay, but let's just wait.
What is min PCT? Oh, what is this min PCT?
This min PCT. Okay, this is. No, it has something with. I'm not sure. Okay, I need to check it. PCT might, okay, thresh use 0.25. It might refer, for example, to, let's say, some fold change or something like that. PCT, I'm not sure if this is something with, let's check. You know, I will not just say something weird, but let's check. So Seurat in general, if you look at the documentation, and okay, let's quickly. Oh, min-PCT. Oh, fine. Okay, now I remember. Yeah, thank you for the suggestion. So min-PCT, it means that, as far as I remember, it takes only the cells for which this gene is present in this many, like minimum percentage of cells where this gene is expressed. So it takes only, you know, this gene. Yes, exactly. Now I remember. Okay. Because this will appear in the output of this function. So let's finish it half more minutes. So in the meanwhile, what I'm telling you, let's just look, for example, at, let's say, Seurat documentation. Yes.
If you have, you define some plus, you need to have plus.
Yes, because it's comparing clusters between themselves.
Okay.
So now, basically, if you look at all these functions, they're all here in a document of, 160 pages. I think it's quite a lot to, to remember all the, but it's a good reference in the case when you need to do some, some very customized analysis. So you can just read this. It's like a book of 160 pages. Yeah. It's a lot. So for example, run t-SNE. We ran this t-SNE, right? So it explains. So basically, not necessarily it's not necessarily to read this this whole book, right, or this whole reference. You can just basically do the ask for the help here. For example, find all markers, right? If you do this thing, you will see that this whole documentation is here in this document. So use either this one or this one, but this is probably easier, right? So now it has a lot of arguments, objects. So this would be glioma, right? Assay. So now, but I think if you have, for example, RNA or ataxy, so you should specify this one. And then which features to use. If it's null, then it's using all the features. Log fold change threshold. All right. So this would be basically threshold on the ratio of expressions. Wilcoxon test. and so forth. So it has, like which test to use. It has so many tests. Like for example, t-test. So okay, if we look at the markers here, so that's what we see here. So basically, this is what these are genes, and you see your p-value, very good one, right? This is the average log-fold change as compared to cluster 0 versus all the other clusters, right? When we find all marker genes, it means that for each cluster, we run the expression against the expression from all the other clusters. So this means that this gene, basically is kind of up related to cluster 0, right? So the gene PTPRD1. So most of the, like a lot of them are from cluster 0, right? So this is where we need the tidyverse for this small function here, for this operator. So basically, it allows us, to take this table, because markers is how many genes it shows us. It shows us 3,000 marker genes, right? It's quite a lot. So that's why we would like to visualize, let's say, top 10 from each cluster. So what happens? Missing. Oh, I don't know what happens. Group by cluster. Where is markers? Okay, just a second, guys. So this is markers, and if we do it like this, it should group it by cluster. Oh yeah, fine. And then it takes, for example, it takes top n by the average log-fold change. Oh, I don't know what happens. Let me try to execute this. And then select gene. It should work, but for some reason it does not. Well, basically this command, okay, just forget about it. What's more, if you didn't have, if you had troubles with tidyverse, basically, you can manipulate directly the table markers, right? Where is it? This one. So it has 3,000 genes, right? You can take, for example, all the genes, and for example, you can see here how many genes, how many marker genes are in each of the clusters, right? And how to compute this? It will be markers versus all other ones. Yeah, there is a possibility in this function to like find markers to compare between specific ones. So now, for example, if we look at this kind of thing, we see that we have 1.2 thousand marker genes for cluster 0, right? 1,000 for cluster 1 and 901 for cluster 2. Okay. Now, let's say we look at some genes here, right? So this is line 194. It's showing us, a violin plot, so basically distribution of gene expression in different clusters, right? Let's say we selected 2 genes here, A to M and QC and KC and Q2. Right? If we look at these 2 genes, we see that basically we have 2 plots in this figure, and it shows us the distribution of gene expression. So basically, most probably A to M is a marker gene for the second cluster. So because we see here, like for example, the first one, it consists of a lot of outliers, but the average expression level is somewhere around 0. The first one is also around 0, and then the third one displays a high level of expression of this gene. So if you know, like, okay, you can just go to some web resources like UniProt or something like that and see what is this gene, what function it has, and then maybe somehow understand what is cell type 2. Okay. So it means like you can find you can take any gene, right? So any marker gene. So for example, let's take this one, the first one here, PTPRZ1. All right. You take this gene, and now if we plot this gene, so what is our expectation is that if its average log fold change is 2.2, most probably it should be upregulated in cell type 0, right? Yeah. Right. So this is really like a very strong marker gene. It has so many okay, fine, but so many cells. This is because it's a lot of cells in cell type 0, right? So that's why we see them all the values there. Okay. Yeah, we can take some other cluster marker genes, and for example, I don't know, what are our markers? For example, if we want to look at markers which have which belong to cluster 1, so we can show them like this, right? So these are marker genes for cluster 1, and now if we want to visualize this one, all right, that's what we have here. This is a marker gene for cluster 1. Okay. Did you guys have, did you do this plot? Okay. I just selected, oh, how I selected this gene?
Yeah, what do you?
I just selected, here, I said that, okay, from the table markers, give me only those genes for which cluster is 1. And then I took one of this.
So for these gene plots, we have to choose some gene from these.
Yeah. Yeah, if you want to visualize it, it's just, it's just one of the.
I want to choose some random
If you want to choose another gene?
Yeah, just some gene not in the list.
Oh, not in the list?
Yeah, top 2.
Oh, okay. Fine, let's do this. So, okay.
Choose some random gene
Let's do this. Okay, so for example, let's get all the genes. Okay, maybe there are like some smarter way of doing it. What I would do is just, let's say, glioma. For example, what is row names? I think this might give us the gene names. Yes, so these are all the genes, right? And or basically, let's take, for example, not all the genes, but from the ones like from variable features, right? Variable features of glioma. So this gives us the list of 2,000 genes. If you remember, we took 2,000, right? So the genes which are marker genes for at least one of the clusters will be what? We take row names of markers. So this would be, the genes. There are slightly more, 2,000 genes, right? But let's see what happens. Can we find something which is difference between these two? Row names, markers. Oh, there is there is function set diff. Okay, now we have a bunch of genes. Okay, So this, you understand what I did here. So basically, out of those 2,000 variable genes which we used to do all this analysis, right? I subtracted the genes which are markers for at least one of them, one of the clusters. And so if you take, for example, one of this from here, oh, why you cannot select it? Okay, I can type. So hopefully it will not be, upregulated in one. Well, let's see the plot. Well, it's all zero. Here it shows like a lot of outliers. If you see here, the mean value is all zero. All right. So this is a gene which is not a marker gene. Okay. So let's see. I think this is the last plot. Okay, the last plot here is also like feature plot. So suppose we want to have the following. We want to have the t-SNE plot or PCA plot, and what we want to do is to visualize, like a gene where in which cells it is expressed. So remember, this was our t-SNE plot, right? And these are like the values of expression or the ranges. And so, for example, in this cluster, you see that gene BCAN is, lowly expressed. And then, for example, in this large cluster or here in this piece it's a highly expressed gene, right? So this can also guide you towards what is the cell type, right? You might have some list of candidate genes. For example, you know that, I don't know, like B cells has one like marker gene, or like natural killer cells. You know what genes to expect there. So you could take this plot, instead of BCAN, paste some other one. Like, for example, let's take the SKA3, which was not expressed, like not a marker gene. So what would happen? It's interesting. Oh, you see, only some outlier cells have high expression, and like in the rest of them, it's very low. Right. So this probably, if you look at this gene now, you say that okay, it's probably not a marker. It's not an interesting gene. Or maybe it's interesting only because it appears somewhere, highly expressed in some subgroup of cells. Maybe it's interesting for these cells, but not as interesting as, for example, what was that name? PTP-RZ1. let's check it. Yeah. See. So, for example, here is lowly expressed. Here is highly expressed. Here as well, and so forth. This is like all this nice plus is just like some good things included in Seurat. If you want to do some other plots, you can take, for example, ggplot or any other packages for that and come up with your own plots. But this is, is it like necessary to know? Probably not, but this is a good tool to analyze genes, markers, like clusters, and so on and so forth. Okay, I think we're kind of done with this. So we can have 25 more minutes and go quickly through 10x data sets if you want, guys. Okay, let's go. So, okay. So this was SmartSeq2 Llama dataset. Let's analyze the data one. Now what I have here. You have only one file there. You can remove it, or you can leave it if you want to play afterwards. It's up to you. But it's not very. It's 68 MB. Not much. Okay.
We're doing the PBMC data set now.
Mm-hmm. So for you guys, I really advise you to do the same thing as you did with the first one, because I have this PBMC 4K dataset on my laptop. I'm just copying to this directory. No, it's not. Okay, is it a zip? Oh, yeah, it's a zip. Yeah. But you should unzip it now. Yeah, you should unzip. Yeah, just unzip it to the same QCB single cell. All right. You unzip it here, that would be fine. So okay, I have something. Wait, I have something different. Oh, what happened? Mac is a bit strange. What happened? Okay, probably it's okay. So, okay, you should, for example, have this filtered gene barcode matrices or PBMC 4K. It depends on what you copied. For me, Max and PowerPK is different.
So we have 3 files in that folder.
So if you go to that folder and look at it, you have barcodes, genes, and matrix. So matrix.mtx, this is a kind of, it should be, I assume this is a compressed file. Oh no, it's, okay, it's a specific matrix format. So which probably tells you, it's a sparse format, which doesn't show you the whole matrix as we saw in the case of SmartSeq 2 dataset. But this one, it tells you, some kind of the coordinates and the values. So it's some kind of abbreviated format. And then genes, this would be, for example, the row names. And barcodes, these are basically, the cell barcode sequences. So that's how you name yourself.
To have them all imported into,
Yeah, you don't need to manually do this, but what you do is the following. So here in our script, let's just change the like one line here. So, but don't erase anything. Let's just comment this line 13 here, and instead, what I think is that we can do is probably wait. There should be a function read 10x. So it will be something like that. Read 10x. Okay. And then we should provide the directory. Oh, what happens?
Provide that PBMC 4K.
Yes.
About that other stuff, is it only one argument?
Yeah, I think it should be one argument here. So let me try, and then let's see what happens. So here in my data, read 10x. Okay. Yeah, I think I got it. Yeah, I have it. So basically here you should provide the argument, the directory where the 3 files are stored. So this should be, up to that files, grc h38. Okay. So most probably you will need to have something like PBMC 4K, something like that, because for me it did not copy on Mac. I don't know what happened, but my suggestion is the following: you go to the folder where you have the 3 files, and then specify the path to this folder as an argument to this function.
So what should I use for cells?
Let's not use anything. Yeah. Okay, did you guys load it?
yes, but can I see if I can find that?
Yeah. Did you guys do it? No? Okay, maybe we should do a Zoom recording. Okay, so let me show the slides again. So here you should go to this URL. Huh? Oh, I need to share again. Okay. So yes, if you want to download this PBMC 4K data set, so you should go, you should type this URL in your web browser's command line. Maybe download and not upload. Where he, I don't know where he wants to upload it. No need to upload, you need to download. Okay, hopefully, guys, hopefully this resolves your problem. Now, I assume that most of you, or 99.9% of you already have this. So let's just quickly, we just, execute all the commands because we're almost out of time, so 10 more minutes. So, okay, you should have the, the sparse matrix here. I need probably to share again the RStudio. Okay. Okay, so let's now create, an object without removing any cells or any genes. Okay, so what happens next is that, okay, let's look at the violin plot. Okay, interesting. Yeah, see, there is some variance, for example, in how many features are expressed. For some of them, this is probably a doublet because it has a lot of genes expressed, right? And okay, probably, I don't know, it's probably more or less good. And this is the total count. Again, I assume that this point is the same one as here. So it's not only has a large number of expressed genes, but also total UMI count. So here we are dealing with UMIs already, not TPMs. Okay.
So how should we subset it?
Yeah. Okay. Yeah. Let's think about subsetting. Good question. So okay. I don't know. Let's for example put the threshold on number of feature like 4,000, right? Or 3,000. Let's say 3,000, right? So when we go here to subset, basically we say that number of feature RNA less than, right? Let's say 3,000. Greater than, okay, 1,000. Let's say. But we're throwing a lot of cells. That's all. Okay, let it be, let's say 2,000. No, not 2,000, like 500. Okay, let's keep it 500 and 3,000. Okay. Yeah. So let's keep it like this. Now we are subsetting this. And then let's go through all the steps. Okay, the object is called glioma, right? But who cares? What this object has now is PBMC cells already. We just only changed the one command there.
Maybe now it's more of a leukemia.
Yeah. Okay, what happens? Okay, now some of the commands will fail. Why? Because in some of the commands I used particular cells from that data set. Now we have different barcode names, so that's why you see like this command is failing, so we cannot execute it. But fine. This is it was just some kind of illustration purposes. What we need to do is to normalize the data, right? Again, these two commands will probably fail. Now find variable features, all right? Okay. Like for example, variable feature plot. This will be a plot. We have some warning messages. Okay, something similar to the previous one, right? So now we have more genes, right? But let's say we're selecting also 2,000. Okay, we run PCA. Okay, now PCA is a bit slow. Okay. So what we can do next is, what else? For example, we can visualize loadings, all kinds of things.
Stick with 2,000.
Not necessarily, but I would do this usually. I would usually take 2,000. I think it should be enough. Then, for example, we can. Okay, these are like visualizing of the loadings, right? Like my goal is now to go to t-SNE plot. Okay, so let's go to the t-SNE plot. So let's find clusters. So number of communities 4. Now we know that BMC should have around 8, right? So probably it's not enough. So that's why we don't go to looking at the cells. Let's set this parameter for the resolution. If it's set it to a higher number, we'll probably see higher number of clusters. 13 is too much. Okay, maybe like 0.2 will be good. 7. Okay, more or less good. 7 is fine. So 7 is fine, and then, okay, we skip all these small functions, which are interesting, but not our goal. And let's compute t-SNE. Okay, we have t-SNE, and let's look at it. Sorry, 23 of what? Oh yeah, now it's 26. Fine, we can go back to 23. So what we see here, you know, like nice clusters. 1, 2, 3, 4, 5, 6. Where is 7? This is probably 7. Okay, but it somehow looks, I don't know, like it's something unusual. Like PBMC looks a bit different. So it might be the the effect of our QC. We just chopped off a lot from the data set. So, no, there should be like there should be some groups. For example, B cell. Like for example, what is there like? CD14 monocytes, they go like with CD8 T cells, like in a group. Here we don't know yet which group is what, but I'm assuming that like looking at this picture, this does not really look as PBMC cells. But fine, let's. Yeah, it's nice. It's nice, but we don't know now what cluster is what cell type. So that's why we need also to run differential expression analysis. So this is PCA plot, okay. Let's run macrogenes. So it can take, it should be okay. Number of communities. So if you want to select number of communities, you just change the parameters resolution. Yes, which does not have, any, let's say, any mapping between number of clusters and this parameter, because it's all data set specific. Okay, so it will run for what, for 7 clusters. Hopefully it will not take much time, but we're already like almost running out of our time. I use 0.2. Yeah, but you're at 8? Oh.
Plus or minus 1.
We lost a cell type. This is not good.
There's a lot of cell types out there.
Yeah, I think it was something different in the seed. But this was seed. Oh, wait, this was seed for the t-SNE transformation, so it should not affect your clusters. So probably we have, we're just doing quickly all the things, and probably we have a bit different numbers. That's what I assume.
Higher resolution equals 4.
Yes. And it basically ranges from 0 to 1, I think. This resolution. Any difference between this? No, I think in this I think that this is the terminology which they use here in Seurat. So they call a cluster community. Which is a synonymous thing, right? So this is the way how they call it. Okay, maybe 1 or 2 more minutes. Let's see. So now, like the last thing which I want to do is to visualize, for example, a feature plot. And we know that, for example, some I don't know, like there are some marker genes for specific cell types. We can try to see and look at the plot if we really can distinguish the cell type there. So there are some, for example, IR7 or something like that, some genes which are marker genes in CD, what, 14 or something like that. So let's try to look at the marker genes. Okay. Yeah, this IL7R, this is one of the marker genes in PBMC. I don't remember exactly which cell type, but this is one of the marker genes. So let's try to visualize this one, for example. Okay. And put it into the feature plot. Okay. Now this should be some kind of what? Lymphoid cells, right? Okay. Or there should be like MS4A1. I remember this one for B cell. Okay, you see, this is B cell. Yes. We have one cell type identified. And so on and so forth. You know, okay, if you're not satisfied with some of the clusters appearing like, you know, in two clusters, then probably your clustering was wrong. Or you need to recompute some of the steps. Or go back to the very beginning and then, you know, try to adjust your QC, how much you throw away. Okay, I think it's 4:30, we are done. So I'm probably stopping the recording and stopping the Zoom meeting, right? Or if you have any questions, let's briefly discuss and then wrap up. Oh, this one? Yeah, if you have your data set.
Yeah, you're just one good afternoon away.
Published, already published.
Oh, yeah.
Okay, so fine, I'm shutting down the recording. Thank you all people who are online for listening to this workshop. Okay, thank you, bye.